Welcome to this next video in which we are discussing COPD. Okay, so we're currently in the process of discussing emphysema. We've discussed what emphysema actually is. It's the loss of the elastic connective tissue from the lungs, and this results in hyperinflation for two reasons. Because of the loss of the elastic connective tissue around the alveoli, which prevents them returning to the healthy size, so that stops full expiration, or rather gives a new resting size for the alveoli, and also the loss of elastic connective tissue around the respiratory bronchioles, which means that when you expire they end up collapsing, which traps air that is downstream of them in those spaces, and therefore they can't empty properly, and that leads to hyperinflation of the lungs and reduced tidal volume, and that leads to problems with gas exchange, which leads to breathlessness, and you compensate by breathing faster, tachypnea, and breathing laboriously with the accessory muscles of ventilation, which is called dyspnea. Okay, right, so now what I want to understand is why does this occur? The pathogenesis. Why does the chronic inhalation of noxious gases lead to the loss of the elastic connective tissue from the lungs? Okay, so pathogenesis then, let's begin. So it starts then by inhaling chronically noxious gases, and it really does need to be chronic. You need to be doing this for years and years, decades and decades. Okay, so when you do that, these noxious gases are going to be coming into your pulmonary acini. They're going to be coming into the alveoli here. Now, these noxious gases are going to contain chemicals that are going to be irritating, and they're going to activate cells of the immune system. So what you have to understand is cigarette smoke is full of poisons, full of horrible chemicals, and it's not just cigarette smoke. Coal smoke and wood smoke are also full of poisons, and the immune system can be activated by these poisons. Now, in the alveoli, we've met cells of the immune system that are present there. We've met these alveolar macrophages. These are what are called sentinel cells, and I'll just write that term down. So the alveolar macrophages that we've met are examples of sentinel cells. And what this means is that they are standing guard effectively. They are ready, waiting, and looking, observing, looking for any signs that there is something horrible about, some pathogen or something dangerous, something that's very dangerous to the body around. And if they find such a thing present, they will call for help. They will call for help from the immune system, bring in a whole bunch of troops that will hopefully be able to destroy that dangerous thing. So sentinel cells are all around the body. In all the different tissues of the body you have sentinel cells, and their job is to stand on guard, and if there is evidence that there is something dangerous about, maybe a pathogen, their job is to then activate the immune system. And these alveolar macrophages are examples of sentinel cells in the alveoli. They're not, however, the only examples of sentinel cells in the alveoli. There are also dendritic cells, which are called alveolar dendritic cells, that sample stuff that's in the alveoli and are looking for dangerous stuff. So let me just show you where these alveolar dendritic cells are present. And for this I'll draw another picture of an alveolus. And I will do the alveolus, I think this time in green. Actually, no, not green. I'll pick another colour. We'll do it in this pink colour. So here, this can be my alveolus here. And then like so. And we will have the alveolar epithelial cells here. So here's a type 1 pneumocyte. Here is another type 1 pneumocyte. Another type 1 pneumocyte. Another type 1 pneumocyte. I might just put in a type 2 pneumocyte for variety. And then we've got another type 1 pneumocyte. And then I'll put in the basement membrane as well. So get the thicker pen. Here is the basement membrane, and of course underneath that basement membrane we know what's going to be there. We're going to have connective tissue, particularly elastic connective tissue. Okay, so let's now put on the alveolar macrophage. Let me undo that and get the thinner pen. So here is our alveolar macrophage. There's its nucleus in white there. And now the new type of sentinel cell that I want to add on to this picture is an alveolar dendritic cell. Now these are not just in the alveolar space like the alveolar macrophages. Instead they are underneath the basement membrane of the alveolus, but they have processes, dendrites, that will penetrate through the basement membrane and therefore can sample stuff that's in the alveolar space. So let me draw one of these. So here, this can be one of the dendrites of the dendritic cell. Here's another dendrite of the dendritic cell. Here's another dendrite that will be sampling stuff that's underneath the basement membrane. And this can be another dendrite. So here is the nucleus. Let me just try and finish it up. So this cell here, again in white, this is representing an alveolar dendritic cell. Now, the reason they're called dendritic cells is because they have these processes which are called dendrites. They are nothing to do with neurons. Neurons also have dendrites, but dendrite is just a name for one of these processes coming off like this. This cell is a cell of the immune system. It is not capable of conducting action potentials. It quite simply doesn't have the necessary voltage-gated ion channels in its cell membrane to be able to conduct an action potential. So it's a cell of the immune system. Its name concerns these processes coming off it. It's nothing to do with a neuron. Okay, right, so the idea here is that these processes extend into all sorts of spaces, and again, this is a sentinel cell that's looking for signs of something dangerous, and it's got these processes extending between the gaps in the alveolar epithelium so that it can sample things that are in the alveolar space. And as I say, its job is to look for things that are dangerous, and if it finds things that are dangerous, it's going to activate the immune system, and that's basically the starting point for all of this. You have these sentinel cells in the alveoli. They are looking for things that are dangerous. You inhale dangerous gas full of poisons all the time. What is going to happen? These things are going to be activated, and they're going to be activated for an extremely long time. What does that mean? They're going to do, because of course you're inhaling the noxious gas chronically. So over years and years, these things are going to be activated, and therefore they're going to be calling for help over years and years and years, recruiting the immune system to come and fight these toxic gases that are present within the lungs, and this is what's then going to lead to the loss of elastic connective tissue, the immune response to the noxious gas. So let's just put this in here. So in this sort of mustard yellow colour, we'll have the noxious gas coming in here. So in yellow here, this is representing the inhalation of the noxious gas, and it's coming into this alveolus, and it's going to activate the sentinel cells in ways that aren't fully understood. We don't know how these noxious gases activate the sentinel cells, but we can understand why they're going to activate the sentinel cells. This gas is full of poison. It makes sense, therefore, for the sentinel cells to respond to it, even if we don't understand the intracellular signalling that might underlie them being activated. So what do these sentinel cells do then when they're activated? Well, they release pro-inflammatory mediators to call for the immune system. They want help. They are alarm cells, basically. They set off the alarm. Now, where is help? Help is in the bloodstream. All of the immune system cells and other components of the immune system, proteins, they're all in the bloodstream. So if these sentinel cells are going to get help, they need to get it from the bloodstream. And that means that the microvasculature has to allow this help to come from the bloodstream and enter the tissue fluid around the alveoli. Okay, so this is what inflammation is all about. These sentinel cells are going to release pro-inflammatory mediators that are going to act on the pulmonary capillaries and tell the endothelial cells of the pulmonary capillaries to recruit help from the bloodstream, so to bring in immune cells and immune proteins. And we'll now look at this process. So, overall, they're going to release what we can call pro-inflammatory mediators, and I don't like the black pen. It doesn't show up very well at all against the dark background. So they're going to release pro-inflammatory mediators, and this is the broad term for any sort of chemical that can be released by a cell that will trigger an inflammatory response. And the inflammatory response is the response of the microvasculature that will allow troops from the bloodstream to be recruited to the tissue fluid. So I'll change pen colour because that's an awful colour to be using. So we'll go to this colour, I think, green. So the two major pro-inflammatory mediators that, if you're just going to learn two for inflammation, the two absolutely major ones that every medical student should know the names of, forever shall their names be revered, are tumour necrosis factor alpha, often abbreviated down to TNF alpha, and interleukin 1. Okay, so I'll write out the names in full. So TNF alpha stands for tumour necrosis factor alpha, and if you've ever studied anything in pathology, you will have probably heard of these before, but here they are again, appearing here. So tumour necrosis factor alpha, there are multiple different tumour necrosis factors, alpha is one of them, and IL-1, which stands for interleukin 1. Okay, so as they say, if you're just going to learn two pro-inflammatory mediators, these are the ones to learn. They are the most important ones that sentinel cells can release to trigger the inflammatory response. And both of these molecules are actually proteins, so they'll be synthesized in the cell and then released. Now, there's another term that people often use with regards to pro-inflammatory mediators, and that's the term pro-inflammatory cytokines. So let me just clarify what cytokines means. So a cytokine is a protein molecule that is released by a cell of the immune system and which will affect the behavior of another cell. Okay, so the important things are that it's a protein, so it has to be a pro-inflammatory mediator that's a protein, and it's going to be released by a cell of the immune system. These sentinel cells are cells of the immune system, and it's going to affect the behavior of other cells. These molecules fit that description. They are proteins released by cells of the immune system, and they're going to go and affect the behavior of the endothelial cells of the pulmonary capillaries to induce the inflammatory response. So let me now just move this up here, and we'll now draw some pulmonary capillaries in. So let me just choose a colour, we'll do it in white. So we're now going to have our pulmonary capillaries here, and of course I've shown this a little bit out of scale. This would be much smaller. It's out of scale with my alveolus. It would be much, much smaller, and it would be around here, right next to the alveolus. But for the basis of the picture, I've zoomed in on the pulmonary capillary here, and here we have the endothelial cells lining the pulmonary capillary here, like so. So here's an endothelial cell, here's another endothelial cell, and they will of course be sat on a basement membrane, so I'll draw the basement membrane in here as well. So here's the pulmonary capillary basement membrane, and often the basement membrane of the pulmonary capillary will be pressed right up against the basement membrane of the alveolus, and therefore effectively they're fused into one, and that limits the diffusion distance for gases. So this here then, this is, oops, I've done that mistake again. Let me just rub that out, oops. All that can go. So this is my pulmonary capillary. So what will now happen is my pro-inflammatory cytokines here, tumour necrosis factor alpha and interleukin 1, they'll diffuse over to the endothelial cells here. So both of these will come to the endothelial cells, and they will change the behaviour of the endothelial cells. And what is the change in behaviour that they're going to induce? Well, the behaviour that they're going to produce in these endothelial cells is 1, endothelial cell contraction, and 2, recruitment of white blood cells. So let me just go over those two things that are going to happen. So one of the things that's going to happen is endothelial cell, so EC is just short for endothelial cell, and then contraction. Now what does this mean? This means that the endothelial cells are going to contract, and they're going to withdraw their peripheral edges. So what's going to happen is the endothelial cells are going to change shape, and they're going to get rid of these end bits, so they're going to become much sort of more globular. So I'll show this. So what might happen is if I have an endothelial cell here, it might go from looking like this to looking more like this. This is what I mean when I say endothelial cell contraction, a change in the shape of the endothelial cell. And if you can imagine what would happen if all of these endothelial cells did that, it would open up gaps between the endothelial cells, and I might just draw it on here. So let's say this one is now going to have this shape shown here in orange. This one's going to have this shape in orange, and it's a bit over-exaggerated, but it gets the message across. If all the endothelial cells change shape in this way, contract in this way, it will open up gaps between the endothelial cells, whereas previously the gaps between them were quite tight because the edges were next to each other like this. So open up these gaps between the endothelial cells, and what this allows is it allows fluid from the bloodstream to go into the tissue fluid here, and also, of course, into the alveolar space, but particularly into the tissue fluid, the connective tissue underneath the alveolus, which will, of course, be filled with tissue fluid. So what you're now going to get is an exudate. That's one of the things that you'll get. And this word, exudate, it specifically means fluid from the bloodstream that contains proteins from the bloodstream. So normally, first, starting from the beginning, remember the blood contains lots of things. It contains cells, it contains proteins, and it contains tiny molecules as well, for instance the electrolytes, which are even just ions. So very small things, but the proteins are very large molecules within the bloodstream. And when you open up gaps between the endothelial cells in this way, via endothelial cell contraction, what's now going to be able to happen is proteins within the bloodstream are going to be able to go through those gaps into the extracellular fluid, the tissue fluid over here. The cells aren't going to be able to move through those gaps. The gaps are too small without help for the cells to actually go through on their own. We'll talk about how white blood cells get through in a moment. But proteins certainly now can, and an exudate is a fluid formed from the blood plasma which contains a lot of the proteins that are in the bloodstream. And why is this so important in inflammation? Well, it's because a lot of the proteins in the bloodstream are very good at fighting pathogens, at fighting dangerous things. So, for instance, there are the complement proteins in the bloodstream that are put in there by the liver. They're very good at fighting bacteria and fungi and other pathogens. So don't just think that the troops that you're going to bring in to fight the dangerous thing come in the form of the white blood cells. You're also bringing in these proteins, and that's what endothelial cell contraction does. Okay, right. So that's what endothelial cell contraction is going to achieve. The other major behaviour that's going to be induced in the endothelial cells is leukocyte recruitment. So leukocyte is the fancy word for white blood cell. So this means the recruitment of white blood cells. So as I said previously, white blood cells cannot get through the gaps between the endothelial cells even after endothelial cell contraction has occurred. They require help to get through those gaps, and the help comes from the endothelial cells. So the endothelial cells can bind, they put on proteins onto their cell surface that can bind to white blood cells, and once the white blood cells bound to their surface, they help the white blood cell through the gap. So if I just draw a picture of this, so let's say we have one endothelial cell there, one endothelial cell here. Both of these have contracted, which is why the gap's there. Let's now say we have a white blood cell, which I'll have in yellow here. What will happen is the endothelial cells can bind to the white blood cell, and then they can assist it in getting through this gap, a process known as diapedesis. Okay, so the endothelial cells assist in leukocyte recruitment, the recruitment of white blood cells into the tissue fluid at the site that has the dangerous entity present. Okay, so let's just summarise what we've seen so far then. So we brought in this noxious gas, it has activated our sentinel cells. Our sentinel cells have released these very famous pro-inflammatory cytokines, TNF-alpha and interleukin-1, and these go to the endothelial cells of the pulmonary capillaries and change their behaviour. They make them contract, and they make them start to recruit white blood cells, and this leads to the formation of an exudate along with the recruitment of white blood cells into this connective tissue underneath the alveolus here. Of course, you'll also get some going into the actual alveolus, but the key that we need to know about is it going into the connective tissue underneath the alveolus, and that's where this inflammatory process is going to lead to the destruction of the elastic connective tissue. Okay, so what we have overall just talked about then is inflammation. This is the inflammatory response. The inflammatory response is a response fundamentally of the endothelial cells of the microvasculature blood vessels to pro-inflammatory cytokines, particularly these two, TNF-alpha and interleukin-1, which pretty much do the same thing. They induce these behavioural changes. Now, let me just talk a little bit about which white blood cells are the major white blood cells that are going to be recruited in an inflammatory response. So there are lots of different types of white blood cells. In an inflammatory response, the first type of white blood cell, the two first types of white blood cell that are generally recruited as part of the innate immune response are neutrophils and macrophages, and these are the two absolutely key ones for understanding COPD pathogenesis, at least at our present level of understanding of how COPD develops. So, the two major ones that are recruited in any inflammatory process, this isn't just COPD, are neutrophils and macrophages, and both of these are phagocytes, so they phagocytose pathogens and break them down. So let me just draw pictures of neutrophils and macrophages. So I'll draw a picture of a neutrophil firstly. So neutrophils have a funny picture. They have this classic nucleus that has a strange shape because it has 3 separate lobes. Now they have only one nucleus, don't ever be confused into thinking they have 3 nuclei. However, they often have a nucleus that has 3 separate lobes and then little connections connecting up the lobes. So here is a picture of a neutrophil then, and this is its nucleus here with 1, 2, 3 lobes and these little stalks connecting the lobes. Now they can have nuclei with more lobes than 3, so you can see neutrophils with 4 lobe nuclei or even 5 or 6 lobe nuclei. And for this reason, neutrophils have another name. They're often called polymorphs because of the many different shapes that their nuclei can take. Poly means many, morphs means shape. So polymorphs is another name for neutrophils. So neutrophils, you have a lot of them in the bloodstream, and they're one of the cells that is always brought in inflammatory responses very early on to deal with the dangerous object that is present within the tissue, and they deal with pathogens by phagocytosing them and destroying them. Okay, right. Macrophages, you do not have macrophages present within the bloodstream. Instead, you have a precursor to a macrophage known as a monocyte present within the bloodstream. And monocytes, if I was to draw a picture of a monocyte, it would look like this. So in contrast to neutrophils, where you have many lobed nuclei, monocytes just have the classical nucleus with a single lobe, effectively, hence why they're called monocytes, to highlight their difference from neutrophils. Then once they've actually gone into tissue fluid, they then differentiate into a macrophage and they become much bigger and blobbier, like so. So here's the picture of a macrophage. Okay, so in this inflammatory response then, we're going to get neutrophils recruited and macrophage recruited into the connective tissue underneath the alveoli. Now, what I'd like to add is the fact that you, especially in COPD, get a huge number of neutrophils recruited, and the reason is that the sentinel cells, for reasons that aren't fully understood, in response to the noxious gas, they release pro-inflammatory mediators that call for neutrophils. So let me add some more pro-inflammatory mediators onto our list of mediators that are released. So I'll put these over here. So both the alveolar dendritic cells and the alveolar macrophages, they also release pro-inflammatory mediators, interleukin 8 and one called leukotriene B4. Now interleukin 8 is another pro-inflammatory cytokine. It's another protein that's released by immune cells and which is going to affect the behavior of other cells. Leukotriene B4, LTB4, and I'll write out its full name here. So leukotriene B4 is a lipid molecule. It's derived from arachidonic acid, similar to the prostaglandins. And both of these molecules, interleukin 8 and leukotriene B4, they are what are known as neutrophil chemotactic agents. So I'll write this here. So they are neutrophil chemotactic agents. Chemotactic agents. Now, chemotactic means that they're going to attract neutrophils. So wherever you have lots of interleukin 8 and leukotriene B4, you end up with lots of neutrophils, and it's not completely understood how they actually work to attract neutrophils, but it is known that they do attract lots of neutrophils. So the experimental fact is that in areas where you have a lot of interleukin 8 and leukotriene B4, you end up with a huge number of neutrophils being recruited there, and therefore they're called neutrophil chemotactic agents. And in COPD, in response to this noxious gas, the sentinel cells, as well as releasing these absolutely key ones for starting the inflammatory response, TNF-alpha and interleukin-1, they also release these neutrophil chemotactic agents, which lead to even more neutrophils being recruited to this area than would have been recruited if you just had the TNF-alpha and interleukin-1. So these, if you like, are the basic ones that call for neutrophils and macrophages as the basic cells of the immune system. These are added extras which are going to, in particular, call for lots of neutrophils. So if there's one immune cell to say, if you're asked for an immune cell that is characteristic of COPD, it is the neutrophil. And if you want to add another one on there, it's the macrophages. Neutrophils and macrophages, at our current level of understanding of COPD, they are the two major ones that drive this process. Now, in reality, if you go to more advanced textbooks, you will find that there are lots of other types of immune cells that are recruited to the connective tissue outside of the alveoli in the COPD process. However, it's not really understood how they actually fit into the process of into the disease process. So I'm not going to talk about those. One day, when we understand the pathogenesis better than we currently do, they may be very, very important. However, at our current level of knowledge, and to give a very basic model of how COPD actually occurs, we're just going to focus on the neutrophil and the macrophage. Okay, right. So, neutrophils and macrophages. So, let me summarise where we've got to then now. So, we breathed in the noxious gas. That has activated the sentinel cells, the alveolar macrophage, and the alveolar dendritic cells in the alveoli. And by the way, you can have more than one alveolar dendritic cell in a single alveolus, and more than one alveolar macrophage there. Just for the basis of this picture, I've drawn one of each. They cause inflammation by releasing the classical pro-inflammatory mediators TNF-alpha and interleukin-1, which leads to an inflammatory exudate forming and the recruitment of neutrophils and macrophages as normal. You know, this is just a standard inflammatory response. Now I've given a bit that's very specific to COPD here. The noxious gas induces these sentinel cells to release these neutrophil chemotactic agents, interleukin-8, which is a cytokine, and leukotriene B4, which is a lipid mediator, so you wouldn't call it a cytokine because remember, as the definition of cytokine, it needs to be a protein. And these somehow, for a mechanism that's not completely understood, lead to a huge amount of neutrophils being recruited here. So you're going to end up with lots of neutrophils and also macrophages in the area underneath the alveoli and also in the area underneath the respiratory bronchioles. So it's going to end up in the area where the elastic connective tissue is. You're going to end up with lots of these neutrophils and macrophages. And I will just draw these on here. So onto my picture here, I'm going to add in, here is a neutrophil, like so. And then here, this is a macrophage. Okay, and we'll have a break here. In the next video, we will see how the recruitment of all of these neutrophils and macrophages to this area where the elastic connective tissue is going to lead to the breakdown of that elastic connective tissue.